Welcome to our channel. In this video, we are going to see about transcription in eukaryotes. Transcription is a process through which RNA is synthesized from the DNA template using the enzyme RNA polymerase. The eukaryotic protein coding genes contain the segments like introns as well as exons. Exons are actually coding sequences. Introns are non-coding sequences. Here, in the eukaryotic cell, this gene structure has both introns and exons, but in prokaryotic cell, there is no introns. So, the transcript of these genes is produced as a pre-mRNA. Then, this pre-mRNA is processed in the nucleus, where the introns are removed and the exons are spliced together into the translatable messenger RNA. And this is the process of transcription, and next one is the translation. You see that picture from the DNA messenger RNA synthesized by using the codes present in the DNA. So the red color line is indicating that RNA, messenger RNA actually utilizes the codes present in the DNA. So this is called transcription. Messenger RNA synthesized from the DNA. And next process actually translation. This is actually the central dogma. Transcription, translation process. Next, by using this messenger RNA coding, the proteins are synthesized. So that process actually called a translation. But today we are only going to see about the transcription in the eukaryotes. If you want to see about the transcription in our prokaryotes, you just see that in our video. Okay, in eukaryotes, this is a very complex system. Each class is transcribed by a different RNA polymerase enzyme. But in prokaryotes, only single RNA polymerase enzyme involved in the synthesizing of the messenger RNA process, and three RNA types like tRNA, mRNA, rRNA synthesized by a single RNA polymerase enzyme. But here, polymerase 1 actually transcribes rRNA genes. Polymerase 2 transcribes protein encoding messenger RNA. Like polymerase 3, it transcribes tRNA. So the code is RMT. Polymerase 1, 2, 3. You just remind that RMT. rRNA polymerase 1. mRNA polymerase 2. Then last one tRNA polymerase 3. Okay, this is a step-by-step reaction. Pre-initiation, initiation, promoter region clearance, elongation, termination. Pre-initiation. The initiation of transcription does not require the primer, because in the replication process, DNA replication, it requires primer, the short sequence of the nucleotide. But here for the pre-initiation of the transcription process, there is no need to like a primer, adding of the primer. RNA polymerase simply bind to the DNA, and along with other factors, some initiating factors are involved here. It unwinds the DNA to create an initiation bubble, so that the RNA polymerase has access to the single stranded DNA template. But however, RNA polymerase does require promoter-like sequences or structure, because the promoter is a short sequence where the RNA polymerase first bind in the particular sequences. So then only the process has initiated here. Proximal promoters, core promoters, TATA. TATA. The sequence is TATA. These promoters are found around -30 base pairs to the start site of the transcription. Not all genes have TATA box, and some genes have TATA AT and TATA TA, like that. Initiation. Unlike the prokaryotic RNA polymerase that can bind to your DNA template on its own, eukaryotes require several other proteins called transcription factors. So it first bind to the promoter region and then help recruit the appropriate polymerase enzyme. Actually, the transcription factors only first bind to this promoter sequence, then only the polymerase enzyme attached to here. But in a prokaryotic transcription, simply the RNA polymerase enzyme attached to the promoter sequences. Okay, the completed assembly of transcription factors and RNA polymerase bind to the promoter, it forming transcription pre-initiation complex, simply called PIC, pre-initiation complex. So most extensively studied code promoter element in the eukaryote is a short DNA sequence, that is starter box. It is present in the -25 to -30 base pair upstream of the transcription site. TF2D, that means transcription factor 2, because RNA polymerase 2 enzyme involved in the synthesizing of messenger RNA. Now we are seeing about messenger RNA synthesizing only. So TF2D, transcription factor 2, D. D is the coding protein, that's all. It binds to the Tata box via the TBP, transcription binding protein. 5 more transcription factors also involved here, and then RNA polymerase combine around the Tata box in a series of stages to form the pre-initiation complex. See that in the picture how the initiation complex was formed. So first TF2D actually bind to the Tata box, then only all the transcription factors are binded here. Transcription factor 2H, TF2H, actually it has some specific role because the separating the opposite strands of the double-stranded DNA. Like in the DNA replication, we will know about the enzyme helicase. Helicase has a role in unwinding the double helical structure of the DNA. You just get the clue H, here also H present. So it also has the same mechanism, it's separated as a single strand of the DNA up to some point or some period, okay. So it involved in the separating opposite strands of double-stranded DNA to provide the RNA polymerase access the single-stranded DNA template. Other transcription factors also involved in this transcription are TF2A, B, E, F. So just like A to F, but there is no C and G protein. You just left out the C and G, then other A, B, E, F, H. So all these proteins are involved in the eukaryotic transcription initiation process. See the picture clearly how the transcription initiation factors are attached with this promoter sequence as well as the polymerase enzyme. The red color enzyme is indicating that polymerase enzyme 2. So these factors are holding this polymerase enzyme to the initiating point like promoter sequences. Okay. The next process is the promoter region clearance. After the first bond is synthesized, the RNA polymerase must clear the promoter. During this time, there is a tendency to release the RNA transcript and produce truncated transcripts. This is called abortive initiation and is common for both eukaryotes and prokaryote cell. Once the transcript reaches approximately 23 nucleotides, it no longer slips and elongation can occur. So this is the elongation process. During the RNA elongation, only the single factor remains attached, that is TF2F. It is attached to the RNA polymerase enzyme, but all of the other transcription factors have dissociated from the promoter initiation complex. One strand of DNA known as the template strand or a non-coding strand, it is used as the template. So as the transcription proceeds, the RNA polymerase transfers the template strand and uses a base pairing complementary with the DNA template to create the RNA copy. And next slide, actually I'm going to show some picture. All RNA polymerase travel along the template DNA strand in the 3' to 5' direction. But the newly synthesized strand direction is 5' to 3' direction. You see this picture. This is the RNA polymerase enzyme. The round shape of the structure is RNA polymerase enzyme, and the double helical structure of the DNA is here. And the red color, actually this is a newly made messenger RNA. How the sequences are formed? You just see the picture here, where the T present in the DNA. So the polymerase enzyme add A. Like A present in the DNA, in messenger RNA, newly synthesized messenger RNA, it will add U because it will not add T. If it is add T, that is a DNA. The major difference between the RNA and DNA is the uracil. the messenger RNA actually has uracil instead of thymine. Other sequences are common, but only one sequence is changed here, that is uracil. So see that picture. The next region is the G, so it will attach C. And next is also G, the blue color. You just see the blue color picture. And G, so it will attach C. T, A, T, A. Likewise, and the complementary sequences of the DNA. So this is messenger RNA. See, it copies the messages present in the DNA only. So the red color indicating that newly synthesized messenger RNA. This is the direction of the transcription. But newly synthesized messenger RNA always have the direction of 5' to 3'. And last process is the termination. RNA polymerase II lacks any specific signals or a sequence that direct RNA polymerase II to terminate at specific locations. Actually, termination process in eukaryotic transcription not very clear compared with prokaryotic transcription. The single-stranded messenger RNA molecule that is produced is released from the nucleus and passes through a pore in the nuclear envelope to a ribosome. Actually, it will reach to the cytoplasm. In eukaryotic transcription, it will reach out to the cytoplasm and directly to the ribosome. This is the site of protein synthesis. And before going to this ribosome, it has some processing like mRNA processing. Okay. Thank you for watching.